Hi guys, I'm presenting this lecture as part of my CHEM651 lecture series. This is a survey graduate level biochemistry course. It is more difficult than Chemistry 451 at the undergraduate level, but it's not a specialized course, it's a survey kind of general course, so it's not quite as difficult as a more specialized graduate, maybe 700, 800 level course. With that in mind, the content overlap between graduate level 651, undergraduate level 451, the content overlap is like 85%. So a lot of the slides are the same between them. The main difference is going to be the depth at which I explain them and the depth at which I expect students to understand it and be able to talk about it in an exam. So if you're an undergraduate, you can watch this and it will be a lot of overlap, and if you're particularly curious, it might help you out as well. So this lecture is on protein function. We've looked at the shape of proteins, we've looked at how the chemistry influences the shape, and now we're going to look at, well, what do proteins actually do? And we're going to look at how the chemistry influences what proteins do and how that causes the physiological effects that we observe. Now we can break down proteins into two different categories, or really especially the globular proteins. We can break them down into enzymes and other. And the enzymes are things which bind to something, change it, and then let go again. The other proteins are things which binds to something and then let go again. There isn't that change in between. And so we're going to look at, first of all, actually just the other proteins, because with the enzymes, you've still got to bind to the thing and let go of it. We just have that extra step of something changing in the middle. But let's start out simple and just look at, well, how do they bind with things and then let them go again? What really kind of shapes that interaction? And also the fact that we can measure that interaction, where there are equilibrium and rate constants associated with that, so we can measure that interaction and then move on to the more difficult part of enzymes, where there's something that changes in between. And we're going to use a classic example as a kind of case study of hemoglobin interacting with oxygen, where it grabs it, moves it around, and then lets go of it again. Now these proteins, the learning goals that I'd like to cover in this lecture are really understanding how proteins bind to other things, and those things that they bind to, they could be small molecules such as hemoglobin interacting with oxygen, but they could be, for instance, other proteins. In this case, we're just going to look at binding and then not necessarily changing in between. We'll save that for enzymes. We're going to look at how proteins bind to other things and then let go of them again. Like I say, there are kinetic constraints and equilibrium constraints, constants associated with that. We're going to look at how we measure those and what they mean, and how we represent them graphically and how that gives us information. We're going to discuss the idea that the 3D shape of the protein, which remember is influenced by chemistry, physics, the laws of the universe, how the 3D shape of the protein changes the function of the protein, which causes the biological impact. So what we're seeing is that chemistry changes the biology. The chemistry influences the biology. We're going to talk about that. And then, like I say, we're going to apply that to hemoglobin binding to oxygen and carbon dioxide. Now, like I say, these proteins have got a range of different functions. Some of them are just going to grab onto something and then just let go. Just as storage, just in that same place. Some of them maybe are going to pick it up, move it around, and then drop it off again. And then we have these specialized functions: defense against pathogens, muscle contraction, where it's going to be binding, but that's part of some kind of function, perhaps to get contraction of the muscles or to signal an immune response. And then, like I say, we have this biological catalysis, where the protein is going to bind to something, it's going to change it, and then let go again. Now, we can think about the interaction that's going on as the protein and the ligand. And remember, the ligand doesn't have to be a small molecule like oxygen, it could be another protein, but it just looks like a small molecule in this case. They come together to form a protein-ligand complex. And this is something that's in equilibrium. So it's going to have constants associated with it that's coming up. And we're going to see that they come together and then they break apart. And so the interaction that they make needs to be largely non-covalent, because the covalent bond is going to be harder to break. It's going to be largely non-covalent. So it's going to be things like hydrogen bonds, static attraction, the hydrophobic effect, van der Waals forces, which are putting them together, allowing them to break apart again. Now, you would have discussed previously quite a lot to do with this lock and key model. Now, as is typically the case in chemistry and science, you learn about something and then it later turns out in later classes that's not actually really the full story. Maybe in some cases it's actually just what we thought 50 years ago and it turns out to be completely untrue. Sometimes it's a blend of, well, we thought this was true and it turns out that actually the model was a lot more complicated, and it's kind of a blend of two different things. And that's the case here. You would have heard about the lock and key model before, and that's the idea that a protein and a ligand are perfectly suited to each other, like a lock and a key, which will specifically binds to each other and get that interaction. The wrong key won't fit into the lock, and the wrong substrate won't bind to the protein. That's what we're saying with the lock and key model. And certainly, you know, we do observe that the protein and the ligand do need to be complementary, otherwise they're not going to come together. They're going to be complementary in size, they're going to be complementary in shape, they're going to be complementary in charge, so you can't have two negative things coming together, they're just going to repel, and hydrophobicity as well, where water is going to push certain things together and increase that interaction. And so we do see that already. But it does make the assumption that things always fit perfectly. And we find that that isn't really the case. We actually find that the induced model fit also is very true. And that is the idea that when things bind, like the protein and the ligand bind, they change in order to fit each other better. Both of them will change to fit each other better. And so that increases the binding and the binding energy, and allows for tighter binding of the ligand to the protein. Both the protein and the ligand can and will change, one or the other or both. So they still got to fit fairly well in the first place. It's just that they're going to come together stronger. Now we can put this all together, and we have, like I say, we have the protein and the ligand coming together to form the protein ligand complex, and there are rate constants associated with that. There's the rate of them coming together and the rate of them breaking apart. And by what we mean by rate is that really it's kind of effectively, if I simplify it, effectively just means kind of efficiency of them coming together. It does mean collisions, but collisions don't always mean binding, right? If it collides backwards, it's not gonna bind together. It needs to collide in the right shape, orientation, position, in order to actually cause that interaction to happen. And so the Ka, and notice this is a lowercase k, which is describing rate. An uppercase K would be constant. The lowercase rate, which is communicating that efficiency of them coming together. Ka, association, you're describing, well, what is the, like I say, the efficiency of them coming together, and the Kd is the efficiency of them breaking apart, which is described as well by the bonds with the weakened molecular forces that are kind of keeping them together. How well can this thing hold on to things that it binds to? How well can the protein hold on to its ligand? And that is reflected by the Kd value. And these follow the standard rate kinetics that were covered in undergraduate general chemistry. This the forward reaction is typically made up of two things coming together to make this one thing. So the velocity is going to be equal to the rate multiply the concentration of one and the concentration of the other. And that makes sense because you have the efficiency, but the efficiency just tells you, well, you know, if they do collide, well, how often they collide, and if they do collide, what are the odds, what's the chance that they are going to actually form an interaction. But you've actually got to have some of the stuff in the first place, and the more of the stuff, the more likely you are to get those collisions and to get a good collision. So it's dependent on how much stuff you have to collide, multiplied by the efficiency of them colliding. Now, again, I'm oversimplifying because after a certain extent you get crowding effects and things like that, but that's essentially it. And we have the backwards reaction, dissociation, as the first order reaction, where it's just really the amount of this multiplied by the how well it can hold on to its ligand, and that will give you the velocity there to be able to measure that. And I show them as Ka and Kd, the rate of them, the rate associated with them coming together, the rate associated with them breaking apart. But many people refer to that as K on and K off. And I realise I've made a small mistake here, and I've put an uppercase K where that should actually be a lowercase k. And because these are rate constants, they have units. And with a first order reaction, it is measured in just per second, and in a second order reaction is measured in moles per second, per mole per second. And we can actually just take these two rates, and the rate, so efficiency of them coming together, divided by the efficiency of them breaking apart, or the strength and ability of the protein to hold on to its ligand, and that will actually tell you the affinity. You know, how much does it want to come together divided by how much does it want to break apart. And if you divide those together, you divide those against each other, then you're actually going to find out, well, how much, well, you know, what is that overall affinity? And that's really the affinity, that's going to be the affinity constant, the Ka. And the way I try to kind of think of it is, I like to think of it as just being like a postman delivering mail. And you've got the efficiency of the postman to deliver mail and the efficiency of the postman to pick up mail, because they've got to pick it up and they've got to drop it off. But the thing is that how much is actually picked up and dropped off is relative as to how much is in the van, right? The postman is going to have to, how well can he pick up the mail, and then the mail is going to sit in the van, and then he can't deliver mail if he hasn't got any. And the more mail there is to deliver, the more deliveries he's going to make. And you know, the amount of deliveries he has to do multiplied by the efficiency of delivering them is going to be the overall velocity, and vice versa for picking up. You know, the amount of things to pick up multiplied by how well he does pick them up is going to be, well, you know, how fast is he able to fill up the truck overall. And if you think about it, well, how at any one point in time, how full is that truck is going to be a factor of, well, how full can he pick up mail divided by how full does he drop off mail is going to communicate, well, what is kind of the resting level of fullness of the truck. And that is the affinity, which is going to be the Ka, which is coming up, the uppercase K, which is coming up on the next slide. So what we're saying in terms of the protein, take it back to the protein, is saying, well, the amount of the protein which is in the protein ligand complex, how much of it is in this state, is relative to how much it wants to come together, divided by how much it wants to break apart, is going to tell you the resting state of this association. Now, this is, like we say, this is in equilibrium. And the important thing is to remember is that equilibrium doesn't mean stopped, right? An equilibrium means you've got a ratio of products to reactants, and that doesn't mean that the reaction has stopped at that point. It means that the reaction is settled, but it's still constantly going back and forth. Things are constantly breaking apart, coming together, breaking apart, coming together, but this is where it rests. And again, it's kind of like saying with our postman is that the postman's perhaps got a certain efficiency for picking things up, a certain efficiency for dropping things off. And if you just let him just do his job for an infinite amount of time, as long as the concentration of letters doesn't change, the fullness of the van is going to stay the same. It's constantly picking up letters, constantly dropping them off, but if the efficiency isn't changing, the amount of letters there are overall in the system isn't changing, it's just going to sit at a certain level. Maybe the van constantly sits at 20%, and it's constantly in flux where things get dropped off and picked up, but it sits at a level of about 20%. That's what we're communicating with an equilibrium. The protein and the ligand are constantly coming together, breaking apart, coming together, breaking apart. But where does it kind of, where does it kind of sit? And at equilibrium, like I say, it's constantly coming together and breaking apart. And so this is the rate of them coming together is equal to the rate of them breaking apart. They come together as fast as they break apart. And we can break down this reaction to just being like this. Protein plus ligand come together to form a protein ligand complex. That's just what's shown here, but without the graphics. And we can rearrange this in the same way as we've got our K is equal to protein over ligand. This is the concentration. Products over reactant. The K, the equilibrium constant, is equal to the concentration of the product divided by the concentration of the reactant, and that's just communicating a ratio. It's saying, well, in this equilibrium, what is the ratio of product to reactant? Remember, where a number greater than 1 is saying that the reaction prefers product, and so it's going to sit over towards product, or a number less than 1 is going to communicate that the reaction is going to sit more towards not making very much product and having more reactant. And that makes sense. If you've got more product, then P is going to be a bigger number, so K will be a bigger number. If you've got more reactant and less product, R is going to be a bigger number, so K is going to be a smaller number. So it's just communicating a ratio. It's just a number that just tells you, well, which one do you prefer? And that's really all this is, where in this case the product is the complex and the reactants are P and L. PL, PL, which is this here. Forgive me for not using these square brackets in this case. And K is, it just means constant. We're just going to call it Ka. Different from the acid Ka, we've just reused the symbol. But in this case, it's the association constant, and it's a measure of where does the equilibrium sit. And like I say, you can measure it as, well, how much does it like to come together divided by how much does it like to break apart. Or you can work it out by being, well, how much is there of both sides of the equation will actually tell you where it sits, right? If this is like saying you can calculate the fullness of the postman's van by saying, well, what's the efficiency of picking up divided by the efficiency of dropping off letters, or what is the fullness of the van by saying, well, what is the ratio of full to empty van, right? It's like 20% full to 80% empty tells you that it sits more on the side of being more empty. It's going to be like 20 divided by 80, which is going to give you a small number. Remember, and that communicates that it's more on the left towards the reactant side. Another thing to mention as well is that for the vast majority of this class, I'm going to be talking about Ka and later on Kd, and I use the constants. You can also reflect these things in terms of their binding energy. Because of the equation with the ideal gas constant and temperature, which is going to be telling you about collisions, multiplied by the natural log of the association constant, is going to tell you about the binding energy of the reaction using this classic chemistry equation. And we can use the binding energy, and often a lot of papers will talk about the binding energy of a protein and a ligand, or many things in chemistry coming together, where a larger, more negative number is reflecting a stronger association, because you're saying that Ka is going to be a bigger number. Ka is a bigger number, then delta G is going to be a bigger and more negative number. And but in this class, I'm not really going to go into binding energy, but I do want you to be aware of it. But I'm going to largely just stick to Ka, because I really want to keep this more kind of theoretical and get across the concepts rather than going through just the math of it. But I wanted to make you guys aware that binding energy is another way that we measure these things, and it really just means the same thing. So we're going to get down into this a little bit more, and we're going to talk a little bit more about this this equilibrium. So in a biological system, you've got a lot more ligands than you do protein, right? You've got a lot more molecular oxygen, O2, in your body than you do hemoglobin by a significant margin. And so it'd be quite handy to know, well, how full is the particular protein? How full is hemoglobin in the system, in your body? And for instance, when you go to the hospital and they put that little thing on your finger and it's measuring oxygenation, It's measuring the fullness of hemoglobin, actually. That's one of the things it's measuring. And so we want to be able to understand that, because the fullness of the hemoglobin, the saturation, the fullness of the protein, that's going to be an important thing to know. And actually it's going to be relative to the concentration of ligand. The more you supply the protein with ligand, the more full it's going to get. And I use the metaphor of water in a bucket. The more water you put in the bucket, the more full the bucket is. That's all it is. The more oxygen you supply with to hemoglobin, the more oxygen hemoglobin is going to hold on to. But remember, the holding on is transient. It also has a tendency to let go. And so it's kind of like thinking of, well, how full is the bucket, how much water is in the bucket is relative to how much water you put in the bucket, but there's also a hole in the bucket. Excuse me. And you know, the bucket can have a bigger or a smaller hole. And if it's got a bigger hole, it's going to take more water to fill it up. If it's got a smaller hole, it's going to be able to get filled up more easily. So we have to kind of factor in the tendency of the protein to break apart into the equation. So really, if we just think of it just as simply as Y refers to the fullness of the protein, how occupied it is. Often it's given the abbreviation sigma, and in fact I see it given as sigma more often than Y. I actually haven't come across it being Y until this particular textbook, but I'm going to keep with it because I'm going to keep referring it to Y in this case, because it helps with the figures and graphs later on in the slides. But very often actually really it's referred to as sigma. In this case it's Y. It just means the fullness of the protein, where 0 is completely empty, 1 is completely full, and where is it in between? It's really just kind of like percent full, ratio full. And the fullness is really, well, how much of it is occupied divided by what is the total amount. It's like the grade on a paper. You know, it's the grade that you got divided by how many the total number of points available. Points that you got divided by the points available. And really you can think of points available as being the points that you got divided by plus the points that you didn't get. So your score on your test is equal to how many points you got divided by how many points you got plus how many points you didn't get, in order to get kind of a percentage grade. What's the ratio of your grade on that paper? And we can actually rearrange this equation to get this equation. And you may be looking at that and thinking, well, how do you rearrange this to get that they don't look similar at all? It's in the textbook, and under the protein function, they've actually got the steps, how they go through this. But essentially, if I was to break it down, we can just set PL to just being 1. And then if we look at P, well, P is, if we rearrange this equation to make P the subject, and then substitute that into here, and then we factor and simplify, then you get this equation here. But essentially, the main thing you need to know is that what this is communicating is that the fullness of the protein is equal to the concentration of ligand that you supply it with, divided by the concentration of the ligand plus the tendency for it to break apart. So the fullness of hemoglobin is equal to how much oxygen you supply it with, divided by how much it wants to break apart. Or the fullness of a bucket is equal to how much. Now if we break down the terms of this equation here, we see that the ligand is on both sides, so that's going to give us a curve later on when we plot this onto a graph. We also have this 1 over Ka, and that is what's referring to the tendency of the protein to break apart. Where you have 1 over a number, you essentially mean the opposite of. So if association is coming together, it's saying the opposite of association. So the fullness of the protein is equal to how much ligand you supply it with, divided by how much the protein has a tendency to break apart. And that's going to, like I say, give you the ratio of occupied binding sites at a particular concentration of L. If we know the association constant, then we can plot at every different ligand concentration how full that protein is going to be. And because L is on ligand concentrations on both sides of the divide mark, we know that it's going to be both sides of the fraction, we know that it's going to be a curve. We're going to come to that in a moment, but first, like I say, we look at that breaking apart as 1 over coming together, but let's just make it simpler and just use the KD value instead, because 1 over KA is equal to KD and 1 over KD is equal to KA. Those numbers are inverse of each other. And so the KD value is really what it sounds like. It's just the dissociation constant. It's, well, how much does the equation sit on the other side? And it's going to be, rather than K is equal to products over reactants, it's K equal to reactants over products. So a bigger number is saying that it is breaking apart less, and a bigger number is saying that's breaking apart more, and a smaller number is saying that it's breaking apart less. So a bigger number is saying that it's going to be over on this side. Yes, a bigger number is saying it's going to be over on this side and breaking down, and a smaller number is going to say that it's going to be on this side and staying together. And like I say, it's just going to be, we're just going to swap those terms around to get the KD value. A lower KD value is indicating a higher affinity, because a higher Ka value is indicating a higher affinity, and they're inverses of each other. And so we can just take that 1 over Ka and just swap it out for Kd. Just makes it, just cleans things up a little bit. So biochemists usually refer to it as the dissociation constant rather than the inverse of the association constant. Just cleans things up a little bit. And this, these KD values can be a range, a big range, but remember I said that the smaller the value, the less it's breaking apart, so that's saying a tighter binding, and so that is something that is more high affinity, such as avidin binding to biotin, and maybe if you do experiments that use streptavidin, this is the kind of chemistry that you're using, where it has a very, very high affinity, so you can pull things out of solution if they're biotinylated. But something like perhaps the insulin receptor, that binds pretty well. We looked at that a few slides ago in the binding energy associated with that. Back, where did I put it? Back here. The binding energy was about negative 55 kilojoules per mole, which is a strong binding. And indeed, we do see that, yes, it binds very, very strongly. It has a very small KD value. Whereas something like calmodulin binds to calcium, and it binds to it pretty well, but not as strong as, several orders of magnitude less than some of these other things. So this is the kind of range that we're looking at, from these small numbers to very, very, very small numbers of a dissociation constant. And a smaller means tighter binding. Now I'm going to just run through just the metaphor. I was saying, just to make it clear, that the fullness of the thing is equal to how much ligand you supply it with, but you also have to factor in that it's going to be breaking apart. Now, if we were to plot this on a graph, then what we're going to get is something that looks like this, where along the x-axis you've got increasing concentration of ligand, along the y-axis you've got fullness of the protein, and we can and so, you know, according to that equation, as you give it more ligand, y is going to increase. And indeed, we see as we give it more ligand, y does increase. But it's L is on both sides, and so it's going to be a curve. And at some point, the protein is going to get saturated. I mean, it can't get any more full than full. An interesting part of this is if we look at the KD value. Now, remember that the definition of the KD value is the dissociation constant. It is where the equilibrium sits in the dissociation of the protein ligand complex. It communicates how tightly the protein holds onto the ligand. That's how we define it. But if we wanted to quantify it, to give it a number, then the number of KD is equal to the concentration of ligand required to half fill the protein. So if we look at the point at which the protein is half full, look at how much ligand was required for that, that number turns out to be the dissociation constant. And the reason for that, if we just look at the equation, well, if we set y to being 0.5, and if we just arbitrarily set L to being 1, and it doesn't matter. You can set L to be anything. You can set it to be 1, you can set it to be 10,000. We can just set it to a number. It doesn't matter what it is. Well, a number divided by that same number plus something else equals 0.5. Well, that's the same as 1 over 1 plus x equals 0.5. Well, x must be 1 because it's just 1 over 2 equals 0.5. And again, you can set that to any number. If it was 10,000, then this would be 10,000, this would be 10,000, and x would be 10,000. It is L when y equals 0.5, because essentially what you've got is a number divided by double that number equals 0.5. So KD must be equal to L when y is 0.5. This is just interesting, just the way that it turns out. So don't get mixed up. KD value is not defined as the concentration required to get half occupancy, concentration of ligand required to get half protein occupancy. That's how we would quantify it, that's how we would give it a number, but its definition is the dissociation constant. It is where the equilibrium sits in terms of dissociation, and it is communicating how well the protein is holding onto its ligand. Now we're going to move past this theoretical stuff and move into the worked example with hemoglobin binding to oxygen and carbon dioxide in a biological system. We're going to add on a few other concepts on top of that as we go along, things like cooperative binding and allosteric modulation. But and some people refer to modulation as effectors. It's the same thing. We're going to talk about some of that stuff in this example, tacking things on, but we're going to kind of move into this worked example now. So I'm going to define a few things first of all so we can understand the biology of what's going on, so that we can then see how the chemistry is influencing that biology. So first of all, I'm just going to talk about something in chemistry, coordination complex. These are a type of organometallic interaction, where organo referring to organic, so those organic molecules that you did in OChem 1 and 2, so things that contain carbon, oxygen, nitrogen, that kind of stuff. Molecules that contain those things, that's carbon-based chemistry, and the interaction with metals. That's going to form a organometallic compound, and you get these coordination complexes. The classic example is something like EDTA, which perhaps you might use in a research lab as a metal ion chelator. It's also used in medicine to treat people who have got metal poisoning. And what these are is that EDTA is a organic compound, right? I mean, this is just an organic compound, carbon, oxygen, nitrogen, hydrogen, nothing surprising here. But we find that these carboxylic acids, they like to bite down on metals, like so. And these dotted lines are representing coordination bonds. This is an organometallic complex. And I use the term bite down very specifically because the number of times that it bites down is referred to being something dentate. So in this second example, if it bites down 2 times, then it is bidentate, and dentate literally as in to bite. And this has a little bit of a historical relevance towards the disease porphyria and vampirism, which is a story for a whole other day. But it's believed that people who had porphyria had, is kind of in ancient Eastern Europe, perhaps had just this disease where they weren't able to hold on to the metal ions due to a mutation in their hemoglobin or something like that. And that gave them bruising and a pale appearance that turned into the legend of vampires. But it's organometallic compounds that are biting down on metals in order to hold them into place. Now this is important because we'll think about hemoglobin and oxygenation and things like that, thinking about levels of iron in your blood, right? And you need to have need to have iron as an essential nutrient in your body in order to carry around oxygen. And the reason for that is because oxygen doesn't dissolve in gases very well, or it doesn't dissolve in aqueous liquids very well. It is a non-polar molecule. We think of oxygen as being something that's polar, but that's relative to it being bound to hydrogen and carbon. When it's just bound to itself, well, it's just, it's a mirror image, so it's non-polar. There is no pole there. It's the same on either side, so it's non-polar. And non-polar things, they don't dissolve in aqueous solution very well. They don't dissolve in water very well. So how does your oxygen get around in your body? How does it transport around? Only about 1 to 2% of the oxygen that you breathe is actually dissolved in the liquid of your blood. Rather, it is carried. And it is carried because it likes to bind to transition metals, such as iron. But you can't just have iron oxygen complexes floating around in your blood. You need to be able to hold on to that iron, bite down onto it, in order to control that iron and control where that iron that's holding on to the oxygen, control where it goes by biting down onto it with the organometallic complex, which is part of hemoglobin. That organometallic complex, that thing that bites down on the iron, is heme, as in the heme in hemoglobin. It is an organic molecule that we see here. It's based upon the structure of histidine, and histidine quite often will hold on to and chelate metal ions. It's part of this porphyrin ring. It belongs to the family of porphyrins, but this particular one is heme. It can have a variety of different metals in the center. Iron is a very, very common one, and it's present in other molecules such as cytochromes that are in your liver enzymes, in the mitochondria of the electron transport chain. But it bites down on metals because life uses these metals for various chemical reactions, or in this case, for holding on to oxygen to transport that oxygen around. Right? The oxygen doesn't want to dissolve in water, but it does want to hold on to iron, and then the heme holds on to the iron, and then the hemoglobin holds on to the heme. I should mention that heme is a prosthetic group. It is a cofactor. It's a prosthetic group, a cofactor. That's something that an external molecule that binds to a protein to help it do its job. We're going to see that, I think, in the next slide or two. There's a couple of pictures to illustrate that. But like I say, this is the protoporphyrin ring, kind of the basic skeleton structure. That looks like this heme here that is biting onto the iron in 4 different coordinate bonds. They're illustrated there by the red arrow. And but what we do know is that iron likes to form 6 coordinate bonds. So 4 of them interact with the heme, but there's another 2 left over. And if we look at this little picture here, this is kind of like looking at this heme, but if we just, instead of looking at it flat, turn it on its side. And so the black lines down here are representing the heme like this. And you can see the red arrows, those are the same red arrows here. But there's also one above and below. Those are the perpendicular ones, which make the 5th and the 6th coordinate bond with iron. And those 5th and 6th, well, the 5th one is a histidine. Remember, I mentioned a little while ago, histidine likes to form coordinate complexes with iron. There's a histidine sitting behind. So that's perhaps, for example, might be this one here. And oxygen makes up the 6th one. So the 5th coordinate bond is with histidine and ahistidine. It's a very conserved histidine in hemoglobin. And the 6th one is with molecular oxygen O2. We call this the proximal histidine. And it's referred to as the proximal histidine because it is behind the iron, but it's close to and behind the iron. There's another one coming up. If you remember from anatomy and physiology, you've got proximal and distal. Distal is further away. There's another histidine which is involved in this interaction in a way, but it's further away, so we call it the distal one. But the proximal histidine, the one that's close by, that's forming this coordinate bond, this is the proximal histidine. And this proximal histidine is it's part of the actual hemoglobin protein itself. So shown in this kind of peachy orange color is a side-on view of the heme forming the 4 coordinate bonds. It's just kind of showing 2 because it's all in plane. And then the fifth, so this is 1, 2, 3, 4, 5, 6 coordinate bonds formed with iron. And this proximal his is part of the actual hemoglobin itself. And this peachy color heme is a extra molecule that slots inside of heme to help it do its job. It's an extra part, an extra tool, an extra component of the protein that helps it do its job, a cofactor. So this proximal his is going to be controlling the game quite a lot. I don't know if I mentioned it's very well conserved. So there's a lot of different globins, myoglobin, hemoglobin. But there's always a proximal his because it has a very important role in stabilizing the iron. But that stabilizing iron, that's what really is controlling picking up and letting go of oxygen. We're going to explore that more. Now, like I mentioned, here in the red is shown the heme group, and you see it's slotted in between the protein, which is shown in this kind of grey-white color, of a globin, such as myoglobin, hemoglobin, neuroglobin, etc., etc. The cylinders are representing alpha helixes, and they are numbered from A, B, C, D, E, F, G, H. So there are 8 of those, and the loops that connect them are the given like AB. So the loop between helix A and helix B are AB. Starts at the N terminal to go to A, ends at cylinder H, helix H, which is the links to the C terminal. And we see that the hemoglobin likes to nestle inside this little nest of globins, of helixes. And the family of globins, they all look a lot like this. We can see that the proximal his is histidine. It's, in this case, this is histidine number 93. It's the his F8, so that's referring to the 8th amino acid on the F helix. Is the proximal one, so it's behind and close to the iron ion, whereas the distal histidine, that's the one that helps out a little bit, but it's further away. That's why it's called the distal. This is histidine number 64, and it is on helix E, and it's amino acid 7. And that's kind of, it actually kind of acts as like, this distal one acts kind of like as a door that controls access to the front, which is where the interaction with oxygen or carbon monoxide happens. And we're going to look at that in a moment. But they all the globins, they kind of look like this is their common tertiary structure. Now this is what the hemoglobin, this is what the heme looks like. We'll start with this image on the right first of all, actually. So we can see that we're looking at the heme here. We have the proximal his underneath. We've got the 1, 2, 3, 4 coordinate bonds, 5 from the proximal his, 6 from the oxygen. And we see that this distal his is helping stabilize that oxygen and stay bound to the iron. This oxygen likes to bind at an angle to the iron ion, but because it binds at an angle, this histidine helps to stabilize it. That increases the affinity that oxygen has for hemoglobin in general. But an important thing to note, and where we're going to get into this a lot more with the meat of this lecture, is that the affinity that the iron has for the oxygen is relative to its position within the ring. When it is flat and planar in line with the ring, it has a high affinity for oxygen, but this histidine, this proximal his, will actually pull it back slightly out of plane, like we see here on the left. This is pulling it back from where it wants to be in plane, and when it pulls it back like that, it loses affinity. It's not going to have much affinity for oxygen. And you may be thinking, well, why would you not want to have affinity for oxygen? The whole point is to have affinity for oxygen. Well, think about this. The postman is not very good if all he does is pick up letters. Has to drop them off too. And this positioning of the proximal histidine controls whether or not the protein has a high or a low affinity for oxygen. So it can have a high affinity in the lungs and a low affinity at the tissues where it needs to drop it off. We're going to dive into that a little bit more as we go on. First of all, I need to mention as well the way that the pocket is shaped in order to fit for oxygen, but not for other things. Now, what we know is that CO is carbon monoxide, and carbon monoxide, it actually binds to transition metals like iron, again, really well. In fact, it binds to transition metals such as iron about 10 million times better than oxygen. And that's a problem, because then this carbon monoxide is going to bind to all the iron in your hemoglobin, and when it does that, the oxygen can't bind. And so that's what causes asphyxiation, and that's what causes the poisoning by carbon monoxide poisoning. You inhale carbon monoxide, it binds to all of the iron in the heme, in your hemoglobin, and then oxygen can't bind, oxygen can't be transported, and you suffocate, you asphyxiate. And so since it binds so well, that's going to be a problem. Now, fortunately, there's a lot more oxygen than there is carbon monoxide, but still, there is enough carbon monoxide that since it binds 10 million times better, that's going to be a problem. Well, what we notice, you can see, is that it binds straight down. The carbon monoxide binds straight down with the iron, whereas oxygen binds at an angle. And that's part of the reason why it doesn't bind quite as well. But biology's got a solution for that. You see, you could have just a heme holding onto an iron, holding onto an oxygen to transporting around, but then it's just going to pick up loads of carbon monoxide. But by fitting the heme within a protein, you can have the shape of the heme, the hemoglobin, shaped in such a way that there's maybe a door in the way that doesn't let carbon monoxide get in. And so you can decrease the access of the carbon monoxide to the heme, and thus decrease the affinity it has. And indeed, yes, the protein pocket of hemoglobin decreases the affinity of carbon monoxide by about 1,000-fold. Okay, it still binds about 10,000 times better than oxygen, but given that carbon monoxide is now in the atmosphere a much lower concentration, in the environment much lower concentration than oxygen, we can handle that now. If it was binding 10 million times better, then the concentration of carbon monoxide is high enough that it causes problems. When we decrease that affinity by 1,000-fold, then now it's not so much of a problem, and the atmospheric carbon monoxide level is low enough that we can get away with it, and it doesn't become that much of a problem. And the way that happens is that, like I was saying, this distal histidine stabilizes oxygen because of the angle at which the oxygen binds to the iron. But if the carbon monoxide were to bind, it doesn't get that stabilization from the distal his, but also the distal his kind of gets in the way. It actually kind of sterically hinders binding of carbon monoxide, and it kind of blocks it off and decreases its access to the iron, and thus they have a lower affinity for each other. So this is the shape of the protein makes it so that oxygen binds better, carbon monoxide binds worse, even though carbon monoxide actually wants to bind to heme more and actually has a higher affinity for pure heme. The biology solves that problem. Like I mentioned earlier, there is a lot of globins in the globin family. Globin is just the family, but there's different specialized varieties for different biological systems, such as myoglobin, where myo is referring to muscles, and this is the protein that's present in the muscles. It's when you have a steak and you cut open the steak and you see all the red. An amount of that is going to be hemoglobin and blood, but actually the majority is going to be myoglobin. That's actually responsible for the red color, the majority of the red color of muscles. Myoglobin is a monomer, so that means it's just one globin on its own, and it's important as a kind of a sink to hold on to oxygen, and also to help pull that oxygen out of the blood and into the muscle tissues. because remember that oxygen doesn't diffuse well through aqueous solutions of your cells, and so in order to get it from the hemoglobin of your blood and pull it through and into the muscles, it needs a little help to pull that oxygen through, and myoglobin is going to facilitate that, because myoglobin has a high affinity for oxygen, because it's a globin holding onto a heme, holding onto an ion that has a high affinity for oxygen. On the other hand, hemoglobin, that's the one that's in your red blood cells. This one instead is tetrameric. This is 4 globins stuck to each other in order to make a quaternary structure: alpha 1, alpha 2, beta 1, beta 2, where the 2 alphas and the 2 betas are protomers of each other. This is responsible for transport around, so it's not just grabbing it and holding on to it, it's not just for facilitating the diffusion, but it's also to move it from one place to another. We're going to contrast these two, the myoglobin and the hemoglobin, and we're going to contrast them in structure and how that difference in structure leads to their difference in function. There's also as well neuroglobin present in the brain, and cytoglobin present a lot in vascular tissues, which we're not going to really talk about too much. Neuroglobin is quite similar to myoglobin in a way, but this is not really kind of relevant for this lecture. But just for your information, there's a whole bunch of different globins. We'll start out talking about myoglobin, because it's just one. It's easier to talk about, and then we can talk about where it's more complicated, where there's 4 of them, and how the 4 of them is what gives it this extra function. It's a classic globin made up of 8 alpha helixes linked together by these loops, where the heme and the ion it's chelating sits in the middle, nestled in the middle, and the structure is very well conserved between all of the different globins. It's just to mention as well, what we're going to come on to a little bit later on is the idea that when we're measuring, when we're looking at this equation down here, where the fullness of the protein is relative to the amount of ligand that is supplied to it, because we're talking about gases in this case, we're actually going to refer to the concentration of the ligand instead of as the concentration of it, rather as the partial pressure. And this is for 2 reasons. I'll delve into this a little bit deeper in a moment, but according to Henry's law, the concentration of a gas dissolved in a liquid is relative to the partial pressure of it. So they're kind of proxies of each other anyway. The partial pressure and the concentration. So the concentration of the ligand, when the ligand is a gas, is proportional to the partial pressure of that ligand. So partial pressure and concentration are kind of used interchangeably anyway. I'll delve into that in a moment. But also another thing is that clinicians prefer to use the PO2, the partial pressure of oxygen, because it reflects the gradients. The partial pressure of oxygen is going to be greater in the lungs and the arterial vessels, and then the pressure is going to be lower in the venous blood vessels and the tissues. And according to Fick's law, gases will move down a pressure gradient, and so that explains why the oxygen is moving between the high pressure of oxygen in your lungs, and it's going down and down and down as it gets to the way it's actually being pulled into tissues which are less oxygenated. It's because it's moving down this chemical potential gradient described by Fick's law. So it's it actually kind of partial pressure describes the movement of oxygen a little bit better, makes more sense to a clinician. And what we find, a common thing that's used is this CaO2, which is talking about the arterial concentration of oxygen. And that is equal to, this is a clinical biomarker that's used by clinicians, and it's equal to the amount of hemoglobin, the concentration of hemoglobin multiplied by how much the saturation of the hemoglobin, so the Y value of it, multiplied by how much oxygen each hemoglobin can hold, a multiplier for that. So how much you've got how much of it is full, how much can it hold, plus the amount of oxygen dissolved in the blood divided by the affinity, the multiplier for the efficiency of the blood, the oxygen being dissolved in the blood, is going to be the total oxygenation. Remember, about 98% is going to be oxygen is attached to hemoglobin, about 2% is attached to the is dissolved in the blood. But the amount attached to hemoglobin plus amount dissolved in blood is the total arterial concentration of oxygen. But notice that we're using this partial pressure of oxygen, and this is going to be lower as you move away from the lungs and into the more deoxygenated tissues. And according to Fick's law, it's going to describe the ways in which it moves along. And like I say, anyway, partial pressure is proportional to the concentration by Henry's law. And the example I like to use is that when you have a can of soda, before you open that can of soda, there's a lot of CO2 dissolved in the water, dissolved in the water of the soda. But then, if you think about it, it's in a sealed system. The pressure is high. When you open that can of soda, what happens to the pressure of the CO2? It decreases as it's exposed to the universe. And as the pressure of the CO2 decreases, what happens to the concentration of the soda? What happens to the carbonation of it? Right? The soda is at its highest carbonation when it is closed, and thus high pressure. And when you open it, the pressure decreases and the concentration decreases, and it is less carbonated. That's Henry's law. So the pressure of the gas is proportional to how much of it is dissolved in the liquid, and it kind of ties this all together in a package that's really handy for the clinician. And so we can just substitute in, instead of having concentration of oxygen, we just substitute in the partial pressure of the oxygen. And instead of using the KD value, we would use the pressure of the oxygen required to half fill the protein. It means the same thing, really, because of Henry's law. But I just wanted to throw that out there so that you're familiar with these things in case you go on to medical school or something like that. So let's get back into it with myoglobin. So myoglobin has a very high affinity for oxygen. Makes sense, it has to. It needs to grab onto that oxygen, needs to grab it from the blood, and it also needs to hold on to it and to make sure it gets delivered to where it goes. So it has a very high affinity. The problem with it having a high affinity is that it doesn't want to let go. It's going to hold on to it, and it's going to stay held on to it. And this graph here is representing that, again, that y is equal to L over L plus KD. It's the same thing. I'm not going to put it in as partial pressure right now. I'm just going to keep it simple. That's what this graph is showing. And what we can see is, yes, as the pressure, as the concentration increases, the fullness increases. There's a hyperbolic curve. It fills up very quickly because the KD value is very low, reflecting that it likes to hold on to it. And so if we think of just our simple protein plus ligand makes protein ligand, go back to Le Chatelier's principle, what you're saying is that if you increase the concentration of ligand, then this is increasing. And if you, because of Le Chatelier's principle, if it's at equilibrium and you increase this, you're going to end up, the equilibrium is going to work to increase this. So if you increase ligand, you increase complex, which is fullness. So yeah, when you increase ligand, you increase fullness. But it also works the other way, right? If you decrease ligand, then you're going to decrease complex. And Le Chatelier's principle says that if you decrease the concentration of ligand, if you decrease the concentration of the oxygen, then the complex is going to break down. When the complex breaks down, what does PL break down to? P plus L. L is oxygen, so it's, instead of holding on to oxygen, it lets go of oxygen. So what this is reflecting is that it fills up with oxygen really, really quickly, but then if the tissue becomes deoxygenated, it starts to let go of it. And that makes sense. Your myoglobin is going to hold on to oxygen until your muscles need it, and then when your muscles need it, they need it because the concentration has gone down. Since the concentration has gone down, it's going to break apart the complex and let go of the oxygen. But this is going to be, you know, you don't want to wait for your tissues to get kind of partially deoxygenated before you drop things off. You want to increase the efficiency of being able to drop things off. What if you could just drop it off sooner? And that's where hemoglobin comes in. It doesn't have this hyperbolic curve. It has something else instead. The affinity is very high for myoglobin. Well, what if you had a system where the affinity can change? And the affinity is high in the lungs and low in the tissues. That means that it's going to form the complex very readily in the lungs, but the complex is going to break very easily in the tissues. When the complex breaks, that's delivering the oxygen. And that's what hemoglobin does. It changes affinity, so it can pick up an oxygen in the lungs and drop it off in the tissues which need it. And this gives it instead a sigmoidal curve, where you see that as the pressure increases, the saturation increases. Yep, that's still the same, but we see that the pressure needs to be quite high in order to fill it up. But when it does fill up, it fills up and gets near completely full. But then when the pressure drops, the fullness drops off really easily, allowing it to be able to drop off that oxygen more efficiently. And so hemoglobin gets almost as full as myoglobin at the same pressure, but when the pressure drops, it drops off a lot more. And if we're thinking, well, you know, just like slightly deoxygenated tissues, well, the myoglobin is not going to deliver very much oxygen, but the hemoglobin is delivering a lot more. So the occupancy is lower. Occupancy being lower means it's delivered. Just like if you've got a mail truck that's full of mail, if it's full of mail and then a moment later it's only half full of mail, well, that's because it's delivered half the mail. What we have here is that the hemoglobin gets full, and then if we look at it at a lower pressure, it's less full. Well, it's less full because it's let go of some of its oxygen. So it delivers more oxygen without needing the pressure, the concentration to drop off as much. And we could say that the KD value has changed, actually. That affinity has changed. The KD value is low in the lungs, and the KD value is high in the tissues. And you're thinking, hey, wait a minute. The KD, that's a constant. Constant can't change. Well, a constant can change if something else about the protein does change, and it changes its shape. It has a shape that means it has a high affinity in the lungs, and it has a shape that means it has a low affinity at the tissues. And the trick to that is that 3D conformation. We're going to jump into that in a little bit, but can you remember the globins are very well conserved? Can you remember the difference between myoglobin and hemoglobin? What was that key structural difference that is going to be that perhaps is going to be exploited in order to get the different dissociation constants in the different stages? We're going to explore that now. Or we're going to look at, this is just really explaining what I've just said. With myoglobin, it fills up really easy. It doesn't take much ligands to fill it up, but the oxygen needs to drop off a lot more in order to be able to, oxygen needs to decrease by more in order to drop off that oxygen. But with the sigmoidal curve of the hemoglobin, we don't see that as much. The oxygen doesn't need to drop nearly as much in order to start delivering some oxygen. So we see why this happens. We see this, there's a structural reason for this. Let's start at the beginning. Let's just look at a simple globin. If we look at the sequence of all the different globins, myoglobin, hemoglobin, neuroglobin, between a human, a frog, a mouse, a monkey, a cat, a dog, the sequence is actually changes quite a lot. The sequence tends to be quite different between them, but it does lead to the same structure, and that's okay. That happens. That's a part of chemistry, because you could swap a valine for a leucine, and those are two different amino acids, so they've lost sequence similarity, but they code for things that do very similar things. You can swap a glutamic acid for an aspartic acid, an aspartate for a asparagine for a glutamine. You can swap similar amino acids, and the sequence has changed, but it's not going to affect the 3D structure very much because the amino acid belongs to the same class and family, and it's structurally fairly similar. So between the different life and between the different globins, the sequence tends to not be that similar. And we can see that this is reflected by the blue here, which is giving a different, is reflecting whether the amino acid has been swapped for something else and how often it swaps for something else in different globins. You see there's quite a lot of blue, so there's quite a lot of dissimilarity. But look, structure's the same. And yeah, if we overlap all of the different structures between a mouse, a human, a dog, hemoglobin 1, hemoglobin 2, myoglobin, they all overlap and look the same, even though the structures, even though the sequence is different. So the tertiary structure between the globins is all the same. So they're unlikely to have a significantly different physiological role and kind of downstream function because of their structure, their tertiary structure differences, or even primary structure differences. Really, it's going to be something else, and that something else is what we mentioned a few slides ago. It's the quaternary structure. The hemoglobin has got it's tetrameric with 4 globins stuck together, and each globin has got its own heme group. Adult hemoglobin has got 2 alpha chains, 2 beta chains, as I mentioned, and it's the fact that you have 4 of them and their relationship to each other. Myoglobin, there's just 1, and it hasn't got another globin next to it to tell it to change. Hemoglobin's got 4, and they kind of communicate with each other in order to facilitate switching between a high and a low affinity state. It's not really communicate, that's not really right, but I'm just simplifying to get the idea across. We're going to look at what it really is in a moment. So the quaternary structure is that you have these 4 globins interacting with each other, hydrophobic effect, hydrogen bonds, ion pairs, same things that you would expect, these non-covalent interactions. In order, this is just 3 different representations. In order to get this quaternary structure made up of 4 subunits, there's interactions between them, between the alpha and beta subunits interact where they're next to each other or adjacent to each other, where the residues, the amino acids, are going to be forming these attractions such as the hydrophobic effect, hydrogen bonds, ion pairs, and salt bridges are going to be forming these attractions. Remember, one very important thing is that these hydrogen bonds and ion pairs, we can change those quite a lot. We can change their strength quite a lot when you change the pH. When you change the pH, that has a massive, remember when we were talking about amino acids, and we're talking about the ionization of the amino acids, the protonation of the amino acids at different pHs, that's going to completely make or break these types of bonds. So that's going to be really, really important where the environment, i.e. pH, is changing the molecule, the amino acid, which changes the macromolecule, the protein, and then that is changing the function of the thing. These huge macromolecular complexes are going to be impacted by the environment and the small chemistry. Now, like I say, we have this problem. Hemoglobin binds oxygen so well that it doesn't want to let go. Or I guess really we could just say globins want to bind oxygen so well it doesn't want to let go. Solution, you're going to push it, when it's time to let go, push it towards a low affinity state. We're going to look at structurally how that happens, what makes that happen. So the hemoglobin is switches between 2 different states, the T state and the R state. The T state stands for tense and the R state stands for relaxed, but that's really just kind of a really weird name for it, because the protein itself isn't really tense in a tense state and relaxed in a relaxed state. It's actually more relaxed in the tense state and more tense in the relaxed state. But I think it refers to more its interaction with oxygen. In a tense state, its relationship with oxygen is tense. In a relaxed state, its relationship with oxygen is more relaxed. I think it more refers to it as that. And as you might guess from that, when it's tense, it's holding on to the oxygen less. So this is a lower Ka, a higher Kd, whereas in the relaxed state it has a higher Ka and a lower Kd. So in the R state it's holding oxygen more, and in the T state it's holding on to oxygen less. And it's the ability for the hemoglobin to be able to switch between these two states. Is going to be able to switch between being in the relaxed state in the lungs to pick up oxygen and a tense state in the tissues to drop off that oxygen, and shifting the equilibrium in order to facilitate that. There's a few different structural ways in which that happens. Like I said, the T state is tense towards oxygen. It's actually relaxed towards itself. It's actually more stable. And it's stabilized, again, like I said, by these hydrogen bonds, hydrophobic effect, ionic interactions. A classic ionic interaction is between His, Hc3, and lysine, aspartic acid, Fg1. And aspartic acid is going to be negatively charged. Histidine, when it's protonated, is going to be positively charged. This negative and positive are going to attract each other, and it's going to stabilize and hold this attraction is going to stabilize and hold the protein together between them. Remember, Fg1 is referring to the loop in between F and G, and the histidine, Hc3, is talking about amino acid 3 on the helix C of the protein, and we have this interaction between them. But because it's a ionic interaction like this, it's dependent on protonation state, and protonation state is dependent upon pH. So that's going to become more apparent as we look on. If we look at the whole structure of the protein, we've got the 1, 2, 3, 4 subunits: beta 2, alpha 1, alpha 2, beta 1, and these are forming attractions, many static attractions. So this is that one that we were just talking about, the asparagine and aspartic acid and the histidine form the attraction. But there's loads of these ionic attractions which are holding the protein together in this. You can see this topology map. And this again illustrates the idea that just because something isn't next to each other in sequence doesn't mean that it's not going to be next to each other in 3D shape. But these are all various ion pairs, salt bridges, ionic attractions, which are going to be influenced by the pH, which is going to change their protonation state. So that is perhaps going to make and break these bonds, which is going to help it move out of the T state and into the R state. We're going to come across that in a little bit. So what does this look like when we plot it? Well, because we have two different KD values, it means that it's going to have two different types of curve. If it was purely in the T state or purely in the R state, then and it didn't switch between them, it's going to have two different distinct hyperbolic curves. We've got this curve here in the high state, where it takes very little oxygen to fill it up, and we've got this type of curve here in the low affinity state, where it takes a lot more oxygen in order to get it to fill up. And so if we look at these colored bars here, these colored bars are reflecting the range of pressures in various tissues versus the range of pressures, concentration of oxygen in the lungs. And of course, the concentration of oxygen in the lungs is higher. Remember that partial pressure, and it's going to move from a high to a low partial pressure, right? This is why clinicians use partial pressure instead of concentration to explain the flow by Fick's law. So yeah, the high pressure, the high affinity state gets full in the lungs, but it gets half full if it was in the low affinity state. But again, looking at the prompts here, can you guys guess? In the high affinity state, is the T or the R? And in the low affinity state, is it the T or the R? Let you think about it for a moment. In the high affinity state, it is the R state, and the low affinity, it is the T state. And so if the hemoglobin was unable to switch between R and T, and it was just all in R or all in T, maybe you had hemoglobin type A, hemoglobin type R, hemoglobin type T, and it couldn't move between the two of them, then it would look like this curve and this curve respectively. And where in the R state, fantastic, you can get 100% full, but it's not letting go of any of that oxygen. Or in the T state, where, well, it only gets half full in the lungs, so that's not great, it's only getting half full, but at least it delivers some oxygen. But if we look at the difference between here and here, That's the amount of oxygen which it's delivering, right? Because that's how full it gets and that's how empty it gets. This is how full the post fan gets versus how empty the post fan gets. And the post fan gets from 50% full to 25% full, so it's only delivering 25% of letters, the difference between 50 and 25. Well, I mean, yeah, 25% is a decent amount to deliver, but we can do better. We can do better, and the trick is that if it's able to get more full at the point of pickup and still get nice and empty at the site of drop off, then it's going to deliver, it's got a greater efficiency rate amount of delivery. And so the difference between here and here is basically 0. The difference between here and here, if it was all in the T state, is quite, it's something, but not that much. But if we have this middle line represents the fact that hemoglobin doesn't exist all as the R or all as the T state, is in an equilibrium where it flicks between the two states depending upon the environment. And so it gets like 90% full in the tissues, and it gets like in the lungs, and it gets like 50% full in the tissues. And so this is much better than all in the R state, and quite a bit twice as good as better as being all in the T state. So it only gets half full, but that's still pretty good. It's still delivering like a good amount of oxygen, better than if it was just constantly in the T state. So you see this flick in between. And we can learn a lot from this graph actually, and we're going to expand upon it as one of the last things we talk about in this lecture. But notice that we have these hyperbolic curves for all in the T or the R, but the fact that it can transition between them gives it an S-shaped sigmoidal curve. And what we can see is, well, what is the point at which it decreases, and what is the steepness at which it decreases? That tells us about how it transitions from one to the other. And like I say, it's in equilibrium between T and R, and this middle line here is actually reflecting a weighted average of the amount of the hemoglobin where you're measuring, which is in the T state versus the R state. Because in the when it's in the R state, it's going to be like this. When it's in the T state, it's going to be like this. And so what we're saying is that at this point right here, we're saying that it's like 60% in the R state and about 40% in the T state. And so the weighted average is that it's at this point right here. So it's either going to be in the R state or the T state, but the proportion between them controls the position of this line, which is why I call it kind of like a weighted average. Around here, it's almost all in the R state, the equilibrium of T to R, it's almost all in the R state up there in the lungs, whereas a greater proportion of it is in the T state at the tissues, and so the weighted average of how full it is a lower amount given by here. So it doesn't exist kind of as, it exists as both the T, this R and T exists as the two, and this line in between is the weighted average at that particular concentration of oxygen. And like I said, the point and position at which it's quite flat here. This portion here is quite flat, and this portion here is rounding out, but this portion here is steep. And how steep and when it becomes steep, that tells us about how it switches between T to R. What is the mechanism by which it flips from T to R? And notice that's quite steep. And that's reflecting that it's not a gradual change from R to T, it's a very rapid change from R to T. And that tells us about how it changes from R to T. We're going to get to that as one of the last things we talk about. Just to mention that when it does change, what's happening is that these 4 subunits, when it changes between the T and the R state, these 4 subunits are going to slide past each other, rotate. The pocket in the middle, I sometimes refer that as the donut hole, which is here, this donut hole. That is going to narrow in the T state. So the donut hole narrows, and the stabilizing ionic bonds are going to break, and new ones will form instead as it transitions between. So the weak intermolecular forces are going to change as it transitions between the different states in order to stabilize a different state. we're going to talk about start talking about and build upon it more and more, the cooperative effect. The cooperative effect is that when oxygen starts to bind, at first it has trouble binding, but as the oxygen binds, it makes it easier for the protein to bind to more oxygen. Remember, each hemoglobin can bind onto 4 oxygens, and it kind of reflects that perhaps the first 1 or 2 oxygens, they struggle to get onto the protein. But then the third and the fourth, or I guess really more the third, because once you get to the fourth, you're reaching saturation. But it gets easier because it's switched into the high affinity state. Again, we're going to expand upon that more, but that kind of explains what we see here. That at first, it's quite hard to get oxygen to bind, but once it does, it gets quite hard for oxygen to let go at first. But then once it does transition, and then when it does transition, suddenly it drops off. We call that the cooperative effect. Oxygen binding helps more oxygen bind. A small amount of oxygen binds first, and it helps its friends bind. It's kind of like if you're trying to climb over a wall, and you'll help each other. You'll help each other get over the wall, and the first person gets a leg up and gets onto the top of the wall, and then pulls the friends over the wall. It's kind of the first oxygen's binding help the next one's bind even more easily. It's the cooperative effect, helps the protein get more full. We're going to expand upon that more. Now, like I said, these attractions are going to make and break. In the T state, we have this ionic attraction, especially with this His HC3. But when it switches into the R state, the shape of the protein changes. And when the shape of the protein changes, it swivels, it changes, it rotates, it slides. And now the HC3 is in the center instead. So a lot of these interactions get made or broken, particularly this His HC3 and this aspartic acid FG1. Oh, and there's the green circles just illustrating this attraction. Now, what does this do? I mean, how does these subunits moving around, how does that change the affinity? Well, the heme is attached to the proteins. When the protein changes, the heme changes too. Kind of. Remember that proximal His? The proximal His, rather, is more correct to say the proximal His is part of the hemoglobin, it's part of the protein, forming that 5th coordination bond, and it is controlling the position of the ion within the heme. And so in the T state, it's actually pulling. So this is the T state. In the T state, the proximal histidine is actually pulling that ion down out of plane. And when it's out of plane, it can't hold oxygen as well. If it can't hold oxygen as well, it has a lower affinity. Low affinity in the T state, because the position of the whole of the protein has changed, so that the histidine, proximal histidine has changed, so that it is now pulling back on that ion within the heme group, and so it loses its grip on the oxygen. Whereas in the R state, the shape of the 4 subunits of the hemoglobin is in such a way that this histidine is pushed forward a little, helps stabilize the position of the ion, so it's nicely in plane with the heme, and when it's nicely in plane with the heme, it can nicely bind to oxygen, and so has a higher affinity. So the histidine controls the position of the ion within the heme group, and the position of the ion within the heme group controls its affinity. Whereas if it's out of plane, pulled back by histidine in the T state, has a low affinity. When it's in plane, held in place partially by the proximal histidine, it has a high affinity for oxygen in the T and the R states respectively. Now, what controls the shape changing? There's going to be, it's not really signalers, but let's simplify it a little bit and say, well, there's signals which are going to be influencing the shape of the protein, and it is the environment, such as the pH. Think about it. If you go for a run, and you're, let's say you go for a run for half an hour, and your muscles start to ache, what is it that's making your muscles ache? Think about what's that molecule? It's lactic acid. Lactic acid is producing protons, decreasing the pH, increasing the concentration of protons, which means in the area of the muscles which are strained and need oxygen, they've got a higher concentration of protons. That higher concentration of protons is the environment, which is that signal which I'm referring to say, hey, I need oxygen. And so high concentration of protons is going to cause protonation. That protonation is going to cause stabilization of the T state. Stabilization of the T state is that low affinity state, so it lets go of the oxygen. Now these things which are influencing the protein, they're referred to as modulators. Some textbooks call them effectors as well. I'd like to see you guys use the term modulators, but effector isn't incorrect. A protein which can bind to something in order to influence, to bind to a different ligand in order to influence its shape, role, function, influence its binding of another ligand is allosteric. So if you've got a protein that binds to ligand A and ligand B influences the binding to ligand A, that is an allosteric protein, and the thing that's doing, the molecule that's doing the influencing, that is the modulator or effector. For example, the ligand for hemoglobin is oxygen, but protons or CO2 or something we're going to come onto in a moment, BPG, these are also ligands. They also bind to the protein, but the binding of protons to hemoglobin as a ligand influences the binding of the hemoglobin towards its main ligand, oxygen. So the protons are acting as a modulator and the protein is allosteric. It's important to mention as well, allosteric refers to outside of the main binding site, so it's not like a competitive inhibitor. The binding site for protons and the binding site of oxygen are separate to each other, so they are allosteric, they're separate to each other, different regions, but the binding of one influences the other. That's the allosteric modulation. If the two ligands, the modulator and the thing that's being influenced, if they are different, they are heterotrophic. Hetero referring to heterotropic. Hetero referring to being different. So a proton and oxygen are two different things. Oxygen and CO2 are two different things. They are heterotrophic. But remember I said that oxygen binds and helps other oxygens bind. Well, oxygen and oxygen are the same thing, so they are homotrophic. So oxygen binding to pocket 1 makes it easier for oxygen bind to pocket 2. Well, pocket 1 and pocket 2 are two different pockets, so they are allosteric to each other. Even though it's within the same hemoglobin, they are different sites, site A and site B. Binding of oxygen to site A makes it so that oxygen can bind better to site B, so they're allosteric to each other, they're homotrophic, because they are oxygen and oxygen are the same thing. So oxygen is a homotrophic allosteric modulator. Protons are a heterotrophic allosteric modulator. They bind in different places from where the oxygen binds to influence the binding of oxygen. Now this is comes in important because, like I said, it's not just oxygen that gets transported, it's the protons and the CO2 for part of the gas exchange. Now, protons and CO2 are things that are produced by respiring tissues. So that's going to that's handy to act as a signaler. Respiring tissues require oxygen. So that's going to be these are going to be things which are going to help shift it into the T state to give it, to decrease the affinity that oxygen has, that hemoglobin has with the oxygen, and so it's going to drop off the oxygen. So the structural effect of protons and CO2 binding to hemoglobin push it into the T state. So the binding of protons and CO2 is inversely related to binding of oxygen. The more protons and CO2 that bind, the more oxygen is released. This is known as the Boer effect. The Boer effect is that pH and CO2, these concentrations of these things, and then binding, influences the release of oxygen by hemoglobin, the Boer effect. Where if you have hemoglobin, when it's protonated, it doesn't have the oxygen. When it's deprotonated, it does have the oxygen. This is in equilibrium. You bind a proton and it lets go of the oxygen. and the levels of each other are going to control where the equilibrium, or the concentrations of the two things across the equilibrium. And of course this can be represented graphically, where the effect of decreasing pH, a greater concentration of protons, remember, more protons, less oxygen. Less oxygen being held onto more protons, the less full the protein is. They're inversely proportional. So a lower pH means more protons, which means less oxygen held by the hemoglobin. And so what we can see is, let's pick just a partial pressure of 4. We've got pH 7.2, 7.4, 7.6. We see at pH 7.2, that is a low pH, a high concentration. It is like the hemoglobin is like half full. We put the pH up, so decrease the protons, it's a little bit more full. Put the pH up more, so there's even fewer protons, and the protein is more full. So let me just delete these horrible little lines. What we can see is that at, for instance, pH, at a concentration of 4 kilopascals of oxygen, at a low pH with lots of protons, it's going to be less full. Higher pH with fewer protons, a little bit more full. Higher pH still with even fewer protons, even more full. So if we decrease the protons, if we increase the protons, we decrease the fullness of the protein. If we decrease the protons, we increase the fullness of the protein. Remember, increase in pH is a decrease in protons. That double negative gets people sometimes. And we typically reflect this, actually. The way to describe it is that the decreasing pH shifts it to the right, shifts the curve to the right. At the high pH, fewer protons, the curve is here. At a low pH, more protons, the curve is here. So we've shifted the curve to the right. By shifting the curve to the right, we make it so we control where the dip part of the curve is. And if we shift it to the right, then we're making it so that the dip part is steeper at this particular concentration, and thus it is going to be at a lower fullness. Whereas if we have it more to the left, then shift it to the left, then the dip part is away, and so the protein is more full. So we're shifting the curve to the left or the right, which influences the fullness of the protein by changing the local environment, by changing the pH. And so in this way, the tissues that need the most oxygen, the most acidified tissues, are going to be the ones that get the most oxygen, because they're going to shift it more into the T state. And so what does this do? Well, if we go back to this slide from earlier, remember that we're talking about the concentration of protons. Well, in order to form this attraction here, this very key main attraction here, histidine needs to be protonated. If there's not enough protons, for instance, at a higher pH, this histidine is not going to be protonated. At a higher pH, this histidine is not protonated, which means this attraction is not made, which means that the protein is not going to be in the T state. Whereas on the other hand, in the acidic conditions of tissues that need oxygen, this is going to be protonated, which means it's going to form this attraction here, which means it's going to stabilize the T state, which is the low affinity, which means drop off oxygen. So more protons, protonates HisHC3, stabilizes T state, drops off oxygen. In the lungs, where the pH is not quite as acidic because it's not producing all of this lactic acid and bicarbonate and things like that, which we'll get onto in a minute, it's going to shift it more into the R state because the T state is not stabilized because this histidine is not protonated. And this is why we studied a lot to do with the pKa of amino acids, and we studied all that stuff to do with the amino acids and whether or not they're protonated or deprotonated at particular pHs, because the protonation state of amino acids at different pHs influences the attractions that they make, and the attractions that they make change the structure of the protein, and the structure changes its function, low affinity, high affinity function. As well as the the protons binding just from, for instance, from lactic acid. Protons are also actually really mainly produced from carbonic anhydrase, and that is an enzyme present in your vascular system. And what it does is it takes CO2 from respiring tissues and it catalyzes the reaction to produce protons and bicarbonate. This is a major part of your blood buffering system. The CO2 that you breathe is important to make bicarbonate, which is a buffering part of your blood buffering system to maintain your pH. But a high concentration of CO2, such as by highly respiring tissues, is going to increase the concentration of protons through this reaction catalyzed by Ca, carbonic anhydrase. And so a high level of CO2 is going to lead to a high level of protons. Those protons are going to protonate His Hc3, which is going to lead to it shifting more into the T state. So the more CO2, more protons, more in the T state, more dropping off oxygen. CO2 and protons are different from oxygen. They bind outside of its of where the oxygen sites, so they are heterotrophic, allosteric, they're modulators, and because they decrease affinity, they are negative modulators. Whereas that oxygen binding, helping more oxygen bind, is that positive modulator we'll get onto later on. CO2 can also directly bind to hemoglobin, and an amount of CO2 does indeed bind to hemoglobin. I believe it's maybe about 10–15% of is transported in this way. And it will, for example, this amine group here, the CO2 will bind to this amine group, and rather than being NH2 or NH3, it is which would be a neutral positive charge, it's going to be this carbamino instead. So it's got a negative charge, and this negative charge can, where's it gone? This negative charge can help to form attractions, which are going to help stabilize the protein in the T state. So when the hemoglobin binds to CO2, that's going to introduce a negative charge, and that negative charge is going to form an ionic attraction, which is going to stabilize the T state. When there's more CO2, there's more of this negative charge, more of this negative charge, more of this T state, more oxygen gets dropped off, and so increasing concentration of CO2 is going to be proportionally related to the binding of O2 to carbon dioxide. It will also be important for transporting CO2 around the body as well. And then finally, another allosteric negative modulator is BPG. BPG is something that is part of biphosphoglycerate, and it is part of glycolysis. It's a glycolytic intermediate. It was one of the last glycolytic intermediates, and so it's going to be produced by a respiration. This actually binds. You see all of these negative charges. See all of these negative charges? These negative charges will fit into the donut hole of hemoglobin and stabilize the T state. So whereas a higher concentration of BPG is going to push things towards the T state, a lower concentration of BPG is going to push things towards the R state. Because it stabilizes the T state, because there's negative charges, positive charges in here on the protein, and the negative charges of BPG help to stabilize that, push the equilibrium towards the T state. BPG is not something that is released by rapidly respiring tissues that require more oxygen to be dropped off. Because as the concentration of BPG increases, then the T state increases, and thus more oxygen gets dropped off. But this isn't something that's released by the tissues to signal, hey, drop off oxygen here. Actually, rather instead, it's produced by the red blood cells, where the hemoglobin actually is. And it's really the response of the red blood cells and the body to respond to longer term hypoxia. So if you are in an environment that over a longer period of time tends to mean that you have a lower concentration of oxygen, then your red blood cells are going to produce more BPG. That's going to shift things more towards the T state, and it's going to drop off more oxygen overall, over the whole of your body. So if you are chronically low in oxygen, your red blood cells are going to produce more BPG so that more oxygen gets dropped off to compensate for that. It pushes the equilibrium slightly more towards the T state. It shifts the curve a little bit more to the right. It does mean that it picks up less oxygen, but it drops off so much more that it's worth it. We're going to have a look at that in a moment too. In fact, we're going to have a look at that right now. So now, what do these different levels of BPG produced by the red blood cells, what do they actually do? What's the physiological effects that we see? And what am I talking about when we see this systemic kind of long-term hypoxia? Well, hypoxia just refers to a lower level of oxygenation of tissues. And there's a few different ways in which this can occur, but let's use the example of being at a high altitude. When you're up in the mountains at a high altitude, there is a lower concentration of oxygen, and because there's a lower concentration of oxygen, the tissues can't get as oxygenated. The hemoglobin can't pick up as much oxygen. Because it can't pick up as much, it doesn't deliver as much, and so you have a little bit less oxygen delivered to your tissues as normal, and that is hypoxia, a lower oxygenation level. And what that looks like is illustrated in this graph here. Let's start out with the blue line, which is just a normal person who's just gone up to the mountains for a day. So at sea level, at ground level, illustrated by the green bar, you can see that the hemoglobin gets pretty full, and then we'll transition through, ignore the yellow bar for now, we transition through to the purple bar. This is with the tissues. And like we know before, in the site of the lungs, they're going to be the higher concentration of oxygen, and at the site of the tissues, there's less. So at the site of the tissues where there's less, we follow the blue line down, and at the tissues, we're at the steep part of the curve, the sigmoidal curve, and we can see that it's going to deliver oxygen to get to, let's say, around about 60% full. So it hemoglobin picks up oxygen to get, let's say, 90% full. And we just use nice round whole numbers to make the math easier. Picks up oxygen to get about 90% full when you're at sea level, and drops off oxygen to being 60% full. So it drops off about 30% oxygen. About 30% of the hemoglobin is going to transition into the T state and let go of its oxygen respectively. And that's going to cause a normal level of oxygenation. Now, what happens up in the mountains? You have a lower concentration of oxygen available because the atmosphere, and because there's less oxygen. Well, here at the site of up in the mountains, the hemoglobin only gets, let's say, about 85% full. At the blue line, 85% full, and it's going to deliver again till it's about 60% full. So it's only going to drop off about 25% oxygen. So now it hasn't dropped off as much oxygen, it's dropped off 5% less oxygen. And that 5% is going to be between the hemoglobin dropping off 25% oxygen or dropping off 30% oxygen. That 5% difference between them is going to cause hypoxia. But it's not quite enough oxygen in order to keep you going. So what happens instead is that the body will release, the red blood cells will start to produce more BPG. If they produce more BPG, they're going to stabilize the hemoglobin being more in the T state. If they stabilize it being more in the T state, that's going to be just like the Bohr effect that we looked at earlier, where the higher concentration of protons, it pushes the equilibrium more towards the T state, and it shifts the graph across to the right. What this means is that now the orange line is illustrated by the change in the increased BPG that have been released by the red blood cells. And so because it shifted the line across this curve to the right, okay, that does mean that at the site of the in the lungs, in the higher altitude, it doesn't get quite as full. It perhaps only gets 80% full. And you're thinking, hey, wait a minute. Before, without the increased BPG, it was getting 85% full. Now it's getting 80% full. That's going to be even worse. It's going to have even less oxygen. Yes, it does pick up less oxygen, but look at how much more it delivers. Now it gets like 40% full, this orange line. And now if we look at the difference between them, that's actually like 40% oxygen. Again, these are not exact numbers. This is just me reading off the graph to illustrate the point. But now you see that it delivers so much more. The difference between these points of when it's in the increased in the T state and these points when it's in the increased T state is that yes, it does pick up less oxygen, but only a little bit less oxygen. It delivers more oxygen, so much more when it's in the more in the T state, and so it's worth it. So yes, by being stabilized in the T state, you switch the equilibrium to being, you shift the curve to the right, you move the equilibrium to being more in the T state. It does pick up less oxygen, but it delivers, the KD is increased, but it delivers so much more. Delivery is prioritized over saturation. It doesn't get saturated, but you deliver more. Delivery is prioritized. So it compensates for it and it's worth it. And what happens if, for instance, you were to go back up into the down to sea level? For instance, athletes who train up in the mountains, they're going to get their BPG levels increasing as a result of adaptation to the lower concentration of oxygen. So they're going to be like in their body's going to be responding like the orange line. But if we go back down to ground level to compete in the games, well, because they're more in the T states, they pick up at the sea level, because they're more in the T state, they're going to pick up 1 to 2% less oxygen than if they were not treated with the BPG. So they're going to be, yeah, sure, slightly less oxygenated, but now look at the difference in oxygen level, the amount of oxygen that's delivered. It picks up 1 to 2% oxygen, but delivers about 20% more oxygen. So it ends up delivering more oxygen, and it's worth it. So that's why they train up in the mountains, then come back to sea level. It's because the body is going to take a while to adapt back to sea level oxygen levels, and so they're just going to deliver so much more oxygen and hopefully perform better in the games. And like I said, and so what you're doing is that by shifting the curve, you're shifting the point at this makes sense, because you're shifting it at the point where the pickup is where the curve is less steep, and so when you shift it, it doesn't make as much of a difference, right? Especially at sea level. When you shift it to the right, because it's not very steep at these 2 points that I put in the square, it doesn't make that much of a difference when you shift it to the right. But because in the delivery point, that's the steep part of the graph, just slightly shifting it to the right, it makes a big difference. Same, the difference between these two, because it's the steep part, is greater than the difference between this, and which is greater than the difference between this. So it's the hemoglobin, the way in which it switches from one state to the other, and that drastic increase by the cooperative effect of oxygen binding has a greater impact in the delivery region, so you can get away with it. And we can explain this cooperative binding by a couple of different models. Remember, like I was saying, that if you have hemoglobin, it's in the T state, but then oxygen comes along and binds to one subunit, that flicks it into the R state and makes it so that the other oxygens can come in and bind. And so if we were to look at this again, back at this graph, at first, it's quite difficult to get oxygen to bind. You need quite a lot of oxygen to get it to bind. But then once you do get it to bind, so it's hard to get it to bind, but when you do get it to bind, it ramps up and fills up quite quickly until it reaches a saturation point where it doesn't matter that you've got quite a lot of oxygen. It's harder to find a space for that oxygen to go into. It's kind of like saying that you've got a parking lot and there's lots of cars trying to get in that parking lot, but there's only a limited number of spaces, and so the rate of filling up spaces is going to slow down a little bit because there's just too much going on. That's why it kind of flattens out here. But the cooperative effect is that oxygen acts as a positive allosteric homotrophic modulator, where the binding of the first amount of oxygen helps to switch the protein into the R state, which makes it more receptive to picking up more oxygen. Well, how does it switch from the T to the R state? Does it do it all at once, or does it do it in part? And that's explained by these two models. The MWC, Monod's, Wyman, Chanju, or the Koshland, Nemeth, Filmer models, where it's the concerted versus sequential, MWC versus KNF. And in the concerted model, what happens is that the protein is either completely in the T state or completely in the R state. And what happens is that the first oxygen binds, and then it flicks the whole of the protein across to the R state. Or in the KNF model, the sequential model, what happens is that there's intermediate hybrid states in between, where the first oxygen will bind to the protein in the T state. That will change some of the adjacent subunits to being in a hybrid state that are more receptive, and then that will change the other adjacent, final adjacent subunit that is going to get more receptive. There's a picture coming up in a minute. Now, so it's either does the protein change all at once, or does part of the protein change, and as more oxygen binds, more parts of the protein change to become more receptive. So is the protein either in the T, all in the T, or all in the R, or is it made up of the subunits, and some of them can be T and some of them can be R? And it'll convert part by part in the sequential model. And it's actually a mixture of the two. But the MWC, the concerted model, is the one that's more favored. What I mean by a mixture of the two is that both happen to an extent. There's evidence that both happen to an extent, but the concerted model is definitely what happens the majority of the time. And that makes sense when we look at this graph. I've gone back again. Is that what's happening is that this steep bit here, that's the rapid change. Now, if where you have an S-curve that's kind of like this, where it's rapid, that suggests that there's a big change that suddenly happens. Now, if it was more like this, then this is a slow change that happens gradually. That's going to be if it was sequential. But that's not what we see. We don't see that. We see this one. And so it must be an all or nothing sudden change. So it's in the T state, oxygen binds, and poof, it pushes it into the R state all at once. And that's what's reflected here, and that's why we have this advantage of not much change when loading, but big change when dropping off. But there is a certain amount, and that's given by the evidence as well, because we don't really see hybrid states. There are ways to get hybrid states by specific conditions, but they're quite hard to get. They're quite hard to find. They're quite rare, and they've only really been discovered actually quite recently. We didn't really know they existed until more recently. We only really find hemoglobin in the T state or the R state. We rarely find it in the hybrid state, and you have to do some special experimental conditions to get that. Not that it doesn't necessarily exist, that's just what we've observed. But it does. There is evidence that there is a certain amount of that as well. Now, what this looks like is the concerted model is here on the left, the sequential model is here on the right. With the concerted model, circles represent T state, squares represent R state. And so if we start up here, we have that the protein is all in the T state, and here we have it all in the R state. And if we look at the equilibrium, it favors, without oxygen, it favors it being in the T state. That makes sense. It's in the T state, it's not as receptive to oxygen, so it's more unloaded. So the equilibrium favors it being in the T state. But what happens? Look at this this equilibrium. Look at it change down the middle, right? What we're saying is that as in the concerted model, as the protein becomes more loaded with oxygen, it makes it easier to switch into the R state, which makes it easier to get loaded with more oxygen. So that by the time it's got 2 oxygens attached to it, you've got a 50-50 mix of T state and R state. By the time it's got 3 oxygens attached to it, it's mostly in the R state. And 4 oxygens, it's again even more so, almost completely in the R state. And so it's either in the T state or in the R state, and the more oxygen you have, the more it pushes it into the R state to flick and change, and then once it gets there, it makes it easier to load. So it's going to kind of follow this kind of route down, and then as it gets more unloaded, it's going to favor being more in the T state. So it's an all or nothing model. Whereas with the KNF model, and so that's what we actually see, and that's why we have this steep bit. Right? That makes sense. That's where it is entered into this kind of region, is this steepness here. Whereas in the sequential model, it's all going to be in the T state, but when oxygen binds, it's going to flick it, that particular subunit into the R state, and then as more oxygen binds, it makes it so that it's easier for the next oxygen to bind and move into the next state, as we can see by the increasing moving down arrow. As it gets more loaded up with oxygen, it makes it easier to fill up. And sometimes I see this one drawn as being like the squares, and then you have these circles, and you have this kind of like hybrid, but also maybe sometimes you have it drawn like this to illustrate that it's a hybrid. So you have the T state, the R state, and the hybrid state in between, and the neighboring subunits. When oxygen binds, that pushes on the adjacent subunits and puts them into a transition state, which is more receptive to oxygen, that ultimately grabs the oxygen and turns completely into the R state, which then pushes onto the adjacent T states that push it into the hybrid state that make it grab onto more oxygen. And actually, logically, the sequential makes a little bit more sense. And if you think of the mechanism, well, what happens when if we go back a few more slides, what happens when it transitions from the T state to the R state? Well, yeah, the tertiary structure is changing some. The majority of the change is from the R state, from the quaternary structure, but you are getting changes to the tertiary structure as well. And it doesn't just go instantly from one to the other. There has to be some movement as it moves from one to the other, right? And so there is an in-between hybrid state as well. So it is a little bit of both, and we could do a whole lecture about talking about this, but I'm just doing a summary, and it is a little bit of both, but it's more the concerted model, and we say that because we see these two a lot more, and we also see the steep change in the slope, which prioritizes the concerted model. And then just to wrap this up with this cooperative effect of oxygen, well, you can actually plot the cooperative effect and see exactly how it is influencing the binding of one oxygen is influencing the binding of another. Now, if we go back to the equation of just protein plus ligand makes a protein-ligand complex, well, the thing is that how many ligands does hemoglobin have? It has a few, because oxygen is a ligand as a ligand, but oxygen is also a ligand as a modulator. So it actually has multiple ligands. Multiple oxygens bind to hemoglobin, and they influence each other. But the main thing is that multiple ligands bind, and because multiple ligands bind, there are n multiple ligands. And so it can form several different complexes. It's going to be several different ligands are going to form to make several different complexes. And the equation actually looks more like this, because there are multiple ligands. And so when we rearrange it, it looks like this. Now the thing is that if these ligands are influencing each other, then this n to the power of, if these ligands are influencing each other, then maybe n is a bigger number. That is going to make it so that the protein is going to get more full, right? So if oxygen is acting as a positive allosteric modulator, then N is going to be a large positive number, which is going to be saying that as you increase the ligand, it's going to fill up faster and faster and faster because they're helping each other out. Whereas you could also have negative modulation, and you can also have as well where there's no modulation actually, where one ligand binding doesn't influence another ligand binding, and in that case N equals 1. So L is just L. L isn't being multiplied up by anything, and so L binding isn't influencing more L binding because it doesn't multiply up. But if you change it to 1, 2, 3, 4, then suddenly what you're saying is that it is multiplying up, and as you increase L, it gets filled up more quickly. Just a quick thing to notice as well, I forgot to mention, is that the steepness of this curve, like I say, is the cooperative effect, and the cooperative effect is explained quite well with the concerted model, but you can't have a negative cooperation with the concerted model. And we do have negative cooperation in terms of the CO2 and the protons. So that doesn't quite fit unless you flip it the other way around and say that oxygen is a positive modulator for oxygen and protons are a positive modulator for protons, and CO2 and protons are both positive modulators for each other to bind on that side. And you can say, well, it's not necessarily that CO2 is a negative modulator for oxygen. It is, but it's a positive modulator for itself, pushing it more into the T state, and it just so happens being in the T state makes it low receptive for oxygen, but it's actually more that you'll think of it as being high receptive for more CO2 itself. So that the concerted model also makes sense from works that way as well. Whereas the sequential model, you can actually have negative modulation, because one thing, the neighbor can make it so that one thing can make its neighbor more or less receptive by changing the shape of its neighbor to being more or less receptive. But you can work the model to work with concerted as well. Just forgot to mention that one. But sorry, going back to here, what we're saying is that N is really the cooperation factor. How much does a ligand affect another ligand binding? And we can, if we solve for N, we can find out how much, quantify the cooperative effect. Now it's going to give us a curve, and we don't like curves that much. Curves are quite hard to solve for. Instead, it's quite nice if we turn curves into straight lines. And we can turn curves into straight lines just by taking log. So if we just rearrange this equation to this, and then log both sides, you get this equals this multiplied by this minus this. And so what this actually looks like is that if you rearrange this equation and then take log of both sides, you get y equals mx plus b. And it's minus, but it doesn't really matter. And so this is a straight line. So you can convert it into a straight line, and this is referred to as the Hill equation. And then what we can do is that we can solve for n, and then that will tell us the n, which is kind of the multiplication factor, which is the extent of cooperation. What is the how cooperative is our particular ligand for the other ligand? We can quantify it. And if n is 1, then it means that there is no cooperation. L is equal to L. You know, L to the 1 is just L. It doesn't matter. But if n is a number greater than 1, then it means that L to the 2 is going to make it so that it's not just equal to L, it's just making it so that when you add ligand, it multiplies up and fills up faster if you have this cooperation. Whereas if you have n as less than 1, then let's say it's L to the 0.5, well, that's saying that you need more ligand. The ligand is only half as effective. You need more ligand in order to fill up the thing. So that's a negative cooperability. So if we can build an equation, solve for n, we can find out the extent of cooperation. And we can do that in the case of hemoglobin. Whereas what we see for myoglobin, well, the slope is just an NH, is this Hill coefficient. the amount of cooperability. What we see for myoglobin, well, myoglobin is the single subunit where there's nothing that influences the binding of myoglobin. So there's no cooperability. Every time you put on one oxygen, one binds, and it doesn't change its affinity. So that doesn't matter. But what we see with hemoglobin in the red instead, let me switch color pen perhaps to a nice green. What we see with hemoglobin, where we have log of X as being the concentration of oxygen, log Y, which is just the fullness of the protein. Convert that curve that we had before into a straight line. What we see here is the protein in the T state, where you supply it with oxygen, and if you just keep giving it enough oxygen, then yes, it is going to get filled up, but there's no cooperation. It's not filling up fast. Whereas what happens is that it will then flick into the R state, where now it's steeper, and now the steepness is n is equal to 3, the slope m is 3, m or nh or n is 3, and so that's reflecting the cooperation that it fills up faster now that it's flicked into the R state. And then it levels out again as it levels out again as it starts to fill up, kind of reflecting that metaphor that I was saying, where the protein, where you've got a parking lot, and let's say at this point right here, you've got lots of spaces and lots of cars, so it gets filled up fast. Whereas here, you've got lots of cars, but not very many spaces, so it's harder for it to fill up. So it starts to, as it gets full, it starts to fill up as it approaches saturation, because it's harder to find a space in order to get in. If you're driving into an empty parking lot, you can find a space immediately like that. If you drive into a parking lot that's only got one space, you've got to find that space, so it's harder to fill up. So it eventually slows down. But this is reflecting that it's hard to find a space because it's let's say poorly arranged, and then it's easy to find a space because there's plenty of them, there's plenty of you, and it's well arranged. And then finally at the end, well, it's not that it's still well arranged, it's just that it's harder for you to find a space, so it slows down. But you can see this cooperation illustrated and in the hill plot here, and you can solve for the slope to tell you the extent of cooperation. And then finally, I've just got I think maybe like 3 more slides just to summarize just a few more aspects of hemoglobin. We're moving away from the chemistry actually in a roundabout way, and more towards a little bit more of a biological function, switching gears, moving into a different direction just to round off a couple of little bits. First of all, talking about fetal hemoglobin. Fetal hemoglobin is, instead of having the alpha beta subunit, we have this alpha gamma subunit. The gamma subunit actually has a lower affinity for BPG. Since it has a lower affinity for the BPG, it's more in the R state. If it's more in the R state, it has a higher affinity for oxygen. And that's important because the child needs to pull the oxygen from the mother's blood, so it needs to have a greater affinity for oxygen. If it was just like adult hemoglobin, then the mother's hemoglobin and the baby's hemoglobin would have the same affinity for oxygen, and so it'd be harder for the baby to take that oxygen. It'd be harder for it to get filled up. It doesn't have direct access to lungs. Whereas it needs in order to remember that with the oxygen's going to move down in the gradients, and so the fetal hemoglobin needs to have a greater affinity in order to pull the oxygen from the mother in order to get filled up. So that's a fetal hemoglobin. It's one type of hemoglobin with the alpha, the gamma subunits. And also I wanted to mention sickle cell anemia. Sickle cell anemia is a disease which causes, it's observed physiologically in the anatomy, in that the red blood cells will change from these kind of soft circular donuts to having these unusual shapes. And the main thing that I want to talk about is not necessarily the unusual shapes and how that may cause damage to blood vessels and things like that, but rather that what causes the unusual shape. That's the important thing for my class here. And what that is that the shape is caused by the hemoglobin and a genetic polymorphism which changes a part of the structure of hemoglobin. Sometimes when I ask a question on this, people will say, oh well, sickle cell anemia causes the sickle cells. Not so much. The polymorphism causes a change in the hemoglobin, which causes a change in the sickle cells. the main thing to concentrate that I'm interested in my course is talking about the polymorphism and what that does to the protein structure. The impact on the cells and how the cells change shape is actually not important for my class. That's not chemistry. That's the biology. I want to talk about the chemistry. And what we see is this substitution of glu-6 to valine-6. Glutamic acid is a acidic, very polar amino acid. It's going to form ionic attractions with things like water. Valine, on the other hand, is very non-polar. And valine is going to stick to valine by the hydrophobic effect. So on the surface of hemoglobin, on the surface, a glutamic acid, which would normally interact with water, is swapped to a valine in people with sickle cell anemia. There's a few different variations. This is one of the most common polymorphisms in the genetic code. And so what that means, if you've got two of them with this patch is going to stick together. That's going to cause aggregation. That aggregation is going to arrange into fibers. These fibers are going to cause distortions in the shape of the cell, which results in the sickle cell that we observe. But this is the disease here. It's the polymorphism causing the forming of the aggregation and the forming of these threads and strands. Particularly stuck in the T state and unable to get back into the R state. Whereas this doesn't happen with wild type hemoglobin. It doesn't aggregate in this way and doesn't cause these this kind of thing. Important thing to note is that there's actually treatments for sickle cell anemia. Now there's genetic treatments I'm not really as aware of, but in terms of drug treatments, there's actually a drug. So this polymorphism occurs in the beta subunit. And remember that in fetal hemoglobin doesn't have the beta subunit, it's got the gamma instead. So there's a drug which actually causes a change in the expression of proteins, and in the fetus does not express beta hemoglobin and instead expresses gamma hemoglobin in their red blood cells. And then when the child is born, the gamma gets expressed less and the beta gets expressed more, and then it switches over the next few weeks and months until there's very little gamma left at all, and it's all adult beta hemoglobin, alpha beta hemoglobin instead. Well, there's a drug that actually switches off expression of the beta hemoglobin and switches on expression of the gamma hemoglobin in adults, so that they don't contain, so that their hemoglobin doesn't contain the disease form of the beta hemoglobin. Instead, it contains a normal, healthy gamma hemoglobin, which means that you actually switch the adults to expressing fetal hemoglobin, which isn't as efficient, but it's better than these fibers forming that causes distortion of the red blood cells. So it's that's an effective treatment which is in place at the moment. But there's also, I hear genetic treatments are slowly coming through as well. So that's everything I wanted to lecture on. Just a summary of everything and kind of the key points that are going to be important for studying and for the exams is that many proteins are going to bind other molecules, including other proteins. And those other proteins are referred to as ligands. And it's always going to be transient because you want to pick up and let go. When the ligand binds, it's going to have to be complementary because otherwise it wouldn't bind. But you've got to remember as well that once of the induced fit model, once it binds, it's going to change in order to bind better. And again, that's actually kind of explained by the concerted model, right? Because when the oxygen binds, it's going to change it into the R state to make it bind better. So it actually makes sense with the concerted model. This can be modulated, but also proteins can have multiple binding sites, and those binding sites can influence each other. Proteins are flexible, and so their shape is going to change, and that change in shape is going to change its function, perhaps from high affinity to low affinity. And it may change it from being high affinity to low affinity, and that's going to explain the induced fit model as well. And that if you have a multi-subunit protein making a supramolecular complex, then this is a very large molecule as compared to some of the small molecules you may come across in OChem, that some of these subunits may influence other subunits. and that proteins can be regulated, and that's the key thing. That's why they're so cool. That you can switch proteins on and off, high activity, low activity, and this is typically influenced by the outside environment, such as other molecules, modulators, pH, and heat. That's why we learn about these things in the previous modules, and that is really the adaptation to the environment, which causes the downstream effect, and that causes the physiology that we observe. That's everything that I wanted to talk about, and if you have any questions, I hope you'll reach out to me. And